8.3, we're looking at the second form of cell division, meiosis. With meiosis, this is going to begin with two parent cells and result in new cells that are somewhat like the mom and somewhat like the dad. So cells will prepare by going through interphase. And instead of like mitosis, going through the cycle once, basically it's going to go through it twice. And this is going to result in 4 haploid cells. And this is going to occur in the testes and the ovaries. So for meiosis 1, we're going to go through prophase, metaphase, anaphase, telophase, similar to mitosis, but these are all going to be phase 1. Prophase 1, the DNA is going to coil up into the chromosomes. Spindle fibers are going to appear. We're again going to have those pairs of homologous chromosomes, now called tetrads. One thing that's going to occur is called synapsis. So there are different sections on the chromosomes, and they're going to be identical on each of the chromosomes, but they will be switching. So this section B here might switch with this section B here, or the D's might switch. And this is going to allow for genetic variation. And so this is called crossing over. So let's say that we've got the pink and the tan. They're going to swap some of their genetic material, and now we have different chromosomes than what we started with. Metaphase is going to occur. So after we've swapped some of that genetic information, we're going to line up, and then again, the spindle fibers will pull those to separate sides of the cell during anaphase. So now we are going to result in 2 cells with different genetic material already than the original parent cell. And telophase and cytokinesis 1, where we're pinching off and resulting in 2 cells. So here we now have half the number of chromosomes in the original cell. Now we will go through a second phase of meiosis. All of those same phases that we just went through in meiosis 1, we're going to do again. So prophase, metaphase 2, the chromosomes are going to line up. Anaphase, they're going to be pulled to the opposite poles. And then telophase, they're going to be split into haploids now. So we've got a half of each chromosome that we started with in meiosis 2. And if you notice, and here it's represented by the different colors, we have 4 different possible options for genetic variation. So the offspring of this parent is going to be different from the parent by a possible 4 different options. Gametes are those sex cells that are produced, so whether they're eggs or sperm. Spermatogenesis is when meiosis is involving the sperm, and here we're going to get 4 sperm produced. Oogenesis is when it occurs in the eggs, and instead of getting 4 eggs produced, now we're going to get 3 polar bodies and 1 viable egg. And those polar bodies are smaller than the actual egg that is usable. So if we look at the top, we're dealing with sperm, and the bottom egg. So everything's very much similar until we get to this last portion. 4 viable sperm produced. We have 1 egg and 3 polar bodies. And that egg then can be used for sexual reproduction. So through sexual reproduction, 1 sperm and 1 egg are going to result in offspring that will have half the traits from each of the parents. If we look at this, we can kind of see this reproduction happening in real cells. And so you can see now spindle fibers being formed, the chromosomes being pulled apart and then split into the two cells. So if we compare mitosis with meiosis, mitosis, you see each phase happens once and the offspring are identical to the original parent. Meiosis, we go through two phases and the offspring are half of the traits from the parent cell. And here we have 4 haploid cells produced, whereas mitosis we have 2 diploid cells.